Next, we have Dr. Yujiro Ichikawa. He has been working on this topic from the Center for Environmental Science in Saitama. He's been looking at this issue for quite some time. He will be joining us online. Unfortunately, he's not able to make it. But he will be talking about the methods of measuring, like how do we measure the VOCs, what are atmospheric VOCs, and really talking about the case study in Japan. We can also think about it for a minute, like all these industries that we have. If you are burning plastic, for example, or if there are some of these other chemical processes that take place, like how do you measure the emissions when the ambient concentration? How do you isolate certain VOCs? And I hope Dr. Ichikawa will also be able to say a few things about this. Dr. Ichikawa, if you're ready, please.
Thank you
Okay,
for
I
the
think
introduction, Mr. Burt. 皆さん、こんにちは。My name is Yujiro Ichikawa. I am a researcher of atmospheric environmental division, institute called Center for Environmental Science in Saitama, which is located in Japan. And first of all, I would like to thank all the staff for providing me this wonderful opportunity to speak. Today I will briefly introduce how atmospheric VOCs are collected and measured, and a case study in Japan. And here's the presentation outline. And I have prepared 3 topics. The first one will be a brief description of VOC and photochemical air pollution. Second one, methodology for VOC collection and chemical analysis. And the third one will be case study of multi-component VOC observation in Saitama, Japan. Okay, then let's move on to the first topic. I'm sure many of you are aware of VOC emission sources and photochemical pollution, but this is an important part to start with, so I will briefly explain. The icons shown here, like airplanes, washing tank, automobiles, gas leak, painting, spray, combustion, vegetation, all of these are VOC emissions. And what I want to emphasize here is that this is just few, and there are a lot more and more sources of VOCs than shown here. And that is one of the main reasons that makes VOC control really difficult. And when the VOC are released into the atmosphere, and plus with a certain amount of nitrogen oxide, plus when there's sunlight and UV lights are exposed to these substances, photochemical reaction could occur, which leads to formation of photochemical ozone and particulate matter like PM2.5. And we all know that these substances are harmful to a human being. And this is the stream of the formation pathway of photochemical ozone. However, I won't go into details to explain this reaction. I just want people to know that the VOC shown in this yellow character right here would be the key for this reaction. And in other words, photochemical ozone formation is triggered by VOC. So it is really important to understand the atmospheric concentration and behavior of VOC. Okay, and this is the first part, and now I will move on to the second topic. Here I will talk about how we collect VOC and how we measure VOC. And this is a slide about the sampling methodology. When you think of VOC sampling, I would say the first choice would be the canister or solvent tube. And you can even see how to use them in the EPA manuals, which are available from EPA website. And here I have written the pros and cons of canister and solvent tubes in a table style. And pros are written in red character, and cons are written in blue character. And I need to say that these pros and cons are my personal thought based on my experience. And I will start with the canister. The first one will be first pros for canister will be the samples are reanalyzable. You can analyze as many as you want until you could inject a sample to analytical equipment. And the second one will be no pump is required because when you use canister for sampling, you will connect to the passive sampler on the top, so which you don't need a power supply. And I would say that it is suitable for a wide range of VOC analytes, and it is suitable for long time sampling, maybe about from 12 hours to about a week. And cons is that I think it's preparation time. Before collecting samples, you will need to condition canister. In other words, cleaning the canister. And before analyzing, you need to dilute the canister. And these procedure will take pretty much time. And talking about 6-liter canister, because it is generally used, are the physical size is about the size of a soccer ball, so it's quite large, and it's a little bit heavy. And the price is about $10 to $15 for 6-liter canister, and 15 to 20 $100 for a passive sampler. So just getting one set of them will be pretty expensive. Okay, then I will move on to solvent tube. The solvent tube preparation time. For preparation, you will just need to condition the solvent tube. So this means that it takes less time than canister. And this is one I personally like about solvent tube is that their size is really small, and the weight is really light. So you can carry around really easily. And it is suitable for short time sampling. I would say from 30 minutes to maybe about a day. And the price is about $150 per tube. And cons is that the types of solvent tubes there are so many. So you need to choose the appropriate one depending on what kind of VOC component you want to measure. And pump is required, so you will need a power supply. And basically, solvent tubes are not reanalyzable, so you need to take special care for it, and special care for collecting and then analyzing solvent tubes. And to summarize the slide here, well, these canister and solvent tubes methods, they have their pros and cons. So it is important to use them according to the situation and what you want to measure or what you want to do. And okay, this slide shows the canister samples, how canister samples are analyzed to determine VOC components. And this is example procedure conducted in my lab. First, we mount the canister on the auto sampler, and then the VOC samples will be sent to the connected VOC preconcentrator. And to describe what kind of operations are conducted at this preconcentrator, I wrote the procedure steps in the bottom of this slide. And the preconcentrator accommodates 3 traps, 1, 2, 3. And as for step 1, the VOC sample from canister will pass through cooled trap 1 and trap 2. And the situation here is that water remains in trap 1. And most of the VOC components will remain in trap 2. And water is something that we want to remove because it will cause negative effects to GC analysis, such as peak separation and sensitivity. So it is really good to be eliminated here. And as for step 2, the VOC components are present in trap 2. So we heat here, and VOC components will move to trap 3. And trap 3 is cooled down to about minus 160 degrees Celsius or lower. And as for step 3, trap 3 is rapidly heated, and the VOC components will be sent to GC for analysis. And the GC system in our lab is a multi-detector, which install a mass spectrometry detector and flame ionization detector. And we use different kind of detectors depending on what kind of VOC components we are analyzing. And okay, this is the slide how the solvent tubes are analyzed in my lab. As you can see the picture, we use the thermal desorption GCMS system. And the bottom left figure shows the procedure steps performed in this system. And as step 1, the solvent tube is highly heated while helium gas flows from the left side in this picture. And then we go to step 2. Step 2 is the VOC components will be desorbed from the solvent tube, and it flows down to the valve right here. And some amount of VOC, the sample will be discarded, but some will go into the built-in tube, which will be cooled to about 5 degrees in order to trap VOC components right here. And for step 3, helium gas flows into this built-in tube to remove water and carbon dioxide. And for step 4, VOC components are, this built-in tube is heated and helium gas flows from the opposite side, and VOC components will be desorbed from this built-in tube, and it goes back to the valve again, and the sample will be injected to GC column and will be detected by MS detector. And the chromatogram chart you see on the right side right here is the result of biogenic VOC, which is the research I am currently working on. And maybe I would like to introduce the biogenic VOC study at another time. And finally, last but not least, case study of multi-component VOC observation in Saitama, Japan. Our lab is conducting observation study of a multi-component VOC, and the objective of this observation is to understand the dynamics of VOC components that contribute to photochemical ozone production. And 100 components of VOC are monitored at 4 sites in Saitama Prefecture from May to September each year. And the reason why we conduct the observation from May to September is that the ozone concentration usually gets high during this period in Japan. And as you see from the map, the map at the bottom, Saitama Prefecture is right here. and Tokyo Metropolitan is right here, so it is really close. It's adjacent, and the four observation sites include one urban area, which will be denoted as UA, and two suburban areas, which will be denoted as SA1 and SA2, and one rural area denoted as RA. And one of the objective of this observation is to monitor a large number of VOC components. So we decided to use canister because it is suitable for a wide range of VOC analytes. And the VOC components observed here are 27 alkanes, 17 aromatics, 11 fluorocarbons, 9 alkenes, 9 aldehydes, 22 halide, 1 alkyne, 3 ketones, and 1 others. And the total of components will be 100. And I have to exclude from today's talk, but aldehydes and ketones were analyzed using LCPDA and LCMS because the sensitivity is a lot higher. And maybe I would like to explain those methodology maybe some other time. And this is a chromatogram chart of GCMS FID system. And VOC components of 2 or 3 carbons which have low boiling points are analyzed by GCFID. For instance, like ethane, acetylene, ethylene, acetylene, and propane, so on. These substances are really difficult to separate and detect by GCMS. So we are using GCFID, and other components will be detected by GCMS. And we are obtaining good separation and good accuracy for this chemical analysis condition. And at the VOC project, which will be starting in full scale from next year, I am willing to give advices to have the VOC components analyzed in good shape. So if there's anything I can do, I'll be always happy. And okay, I will move on to the result here. Total VOC trends of total VOC concentration. Total VOC concentration is the sum of the 100 VOC components. And you will see the trends at the 4 sites in line graph from year 2010 to year 2020. And from this figure, I can say that the concentration are low at the rural area than at the other sites. And this is probably due to fewer anthropogenic sources in rural areas. And the concentration of urban area and suburban areas have decreased over the decades. And one possible factor contributing to this downtrend is the effect of air pollution control rule revision, which was set to reinforce the VOC control in year 2006 in Japan. So the previous slide showed that the VOC concentration, especially in urban and suburban areas, have decreased. So what about NOx trend? As I explained a little bit earlier, NOx is another key substances for photochemical ozone. And this is the trend of NOx concentration from year 2000 to 2020 in Saitama. And as you can see, it is clearly showing a significant decrease. The reason for this trend would probably due to the regulations for automobiles and increase of eco-friendly cars like electric vehicles, and conversion to good quality fuels are also likely to have a significant impact. So now we know that VOC and NOx level have decreased. So I think that people would pose this kind of question. How about ozone? And this is a trend of ozone indicating in annual maximum of hourly concentration from 1983 to 2021. And Saitama is shown in green, and places close to Saitama like Tokyo in yellow, Chiba in purple, Kanagawa in blue, and these four average in orange are also shown in this line graph. And we can confirm that there has been a slight downward trend since around 2006. And this is probably due to the effect of Japanese law revision for VOC control, which I mentioned a little bit in the previous slide, and voluntary activities by companies to reduce the use of VOC. However, the quality standard for photochemical ozone in Japan is 60 per billion per hour. So this means that we have still a gap, and we have to reduce a little bit more. And yeah, reduction is will be needed. And in order to further decrease ozone concentration, there is a thing that I can say from the result of our multi-VOC component observation study. And this figure shows the stacked bar chart of VOC component groups at each site from year 2010 to 2020. And you can see that alkanes, which is indicated in green blue bar, and ketones in light green bar are decreasing. However, other groups do not show as large decreases as alkanes and ketones. So I summarize what I want to say from this result. From individual observation of VOC components, we found that there's little or no decrease in alkenes, aldehydes, and aromatics. And for the many components in like alkenes, aldehydes, aromatics, these groups are known to exhibit relatively high photochemical reactivity. So I would say that counter measurements against these VOC groups might be one of the key for further reduction of ozone concentration. And this is one of the conclusion we have reached. And this will be the last slide for today's my talk. From this case study, I would say that it is important to understand the trends of total VOC concentration. However, just monitoring total VOC concentration is not enough to promote VOC countermeasurements. It is necessary to accumulate scientific knowledge on the individual VOC components that differ in source and photochemical reactivity. So to accumulate scientific knowledge, this is where analytical techniques and monitoring of individual VOC components will be needed. So, okay, people who are interested in measuring VOC, I am always happy to share information and knowledge. So feel free to send me a message anytime. Okay, this is it. Thank you for your time.